Inside your body are cells that have stopped dividing, stopped doing their normal job, but somehow refuse to die. Scientists call them senescent cells. Most people know them by a more unsettling name, zombie cells. These cells can build up in tissues as we age, and instead of remaining silent, some release inflammatory signals that disturb nearby healthy cells. For years, researchers have searched for a safe way to remove them. Now, scientists may have discovered a hidden weakness inside these aging cells, one connected to an enzyme, the fats in their membranes, and a destructive process that behaves almost like biological rust. But there is a complication. That weakness may also affect healthy cells nearby. Senescence is not always harmful. When a cell becomes badly damaged, allowing it to keep dividing could spread mutations or increase the risk of cancer. So the cell activates an emergency brake. It permanently stops dividing. This can protect the body in the short term. The problem begins when senescent cells remain in tissues for too long. They can release inflammatory molecules, enzymes, and growth signals into their surroundings. Imagine a damaged factory that has stopped producing anything useful but continues releasing chemical pollution into the neighborhood. That is one reason researchers believe accumulated zombie cells may contribute to tissue decline and age-related disease. To remove them safely, scientists first need to identify something that makes these cells different from healthy ones. Researchers investigated whether zombie cells were vulnerable to a form of regulated cell death called ferroptosis. Unlike apoptosis, which is an organized self-destruct program, ferroptosis happens when fats in the cell membrane become severely damaged through oxidation. Iron-dependent reactions cause damaged lipid molecules to accumulate. This damage can spread across the membrane like rust moving through metal. Healthy cells normally use antioxidant systems to control it, but when those defenses fail, the membrane breaks down and the cell dies. The researchers wanted to know, are senescent cells easier to destroy through ferroptosis than younger cells? The scientists studied human lung fibroblasts grown in the laboratory. They allowed some of the cells to divide repeatedly until they entered senescence. Then they exposed both young and senescent cells to a chemical used to trigger ferroptosis. The result was clear. The zombie cells were much more vulnerable. Something inside them was making their membranes easier to damage. When the researchers examined their lipid metabolism, one enzyme stood out. It was called acid ceramidase. Levels of this enzyme increased as the cells became senescent. Acid ceramidase helps process lipid molecules called ceramides. These molecules are important parts of cell membranes and also participate in signals related to stress, survival, and cell death. In the senescent cells, increased acid ceramidase activity was linked to changes in membrane fats. The cells accumulated more polyunsaturated fatty acids, fats that are especially vulnerable to oxidation. This may explain why zombie cells were more sensitive to ferroptosis. It was as though they had rebuilt their protective walls using more flammable material. To test whether the enzyme was truly responsible, the researchers reduced acid ceramidase. The cells then became more resistant to ferroptosis. That suggested the enzyme was directly influencing their vulnerability. But this also means scientists cannot simply block the enzyme and expect zombie cells to die. Instead, they may need to exploit the weakness created by high acid ceramidase activity. Then researchers found something even more surprising. Zombie cells appeared to make nearby young cells more sensitive to ferroptosis as well. They did this by releasing signals into their surroundings. This is called a paracrine effect. It means an aging cell can influence another cell without directly touching it. So a small group of zombie cells may create a wider zone of vulnerability inside a tissue. That discovery raises an important safety concern. A future treatment designed to destroy senescent cells could also damage nearby healthy cells that have already been sensitized by those signals. This discovery is exciting, but it is not an anti-aging treatment. The research was performed in cells grown in a laboratory. It is not yet shown that targeting acid ceramidase can safely remove zombie cells from animals or humans. Scientists still need to understand how this pathway behaves in real tissues containing many different cell types. They must also determine whether harmful senescent cells can be targeted without removing cells that may still be useful for healing or protection against cancer. The discovery gives researchers a new molecular target, but many questions remain. So have scientists found a way to eliminate zombie cells? Not yet, but they may have discovered something almost as important: a weakness hidden inside the fats that form their membranes. The next challenge is learning how to target that weakness without placing healthy cells at risk. And if researchers solve that problem, future medicine may one day help tissues remain healthier for longer. Subscribe to BioFacts as we explore the discoveries changing what we know about aging, disease, and the human body.